Good day, this is Professor Will, CBMD PhD. Today's Sunday, October 4th, 2020. It is 7:40 and 28 seconds p.m. Eastern Standard Time, and this is a continuation of my microbiological research, so let's hop to it here. Phylogenetic systematic seeks congruence between the lines of descent evolved over time, and the supraspecific taxa described by taxonomists. Prerequisite for the description of a taxon of any rank in the phylogenetic system is the recognition that all members originate from one ancestral form and that homologous traits of the ancestral form are found also in their descendants. The question then remains, which of the several thousand semen tides in a prokaryotic cell are useful for phylogenetic studies? The establishment of a system which is set up to subsume all species must include phylogenetic markers that are ubiquitously distributed, functionally equivalent, and homologous housekeeping molecules. These markers should be homologous apomorphic characters that evolved only once, synaptomorphy, but not by convergence. Homology is the sharing of two taxa, a property derived from the same or equivalent property of the nearest common ancestor. Deciding whether a property is homologous or the product of convergence has been the greatest problem, and one that could have been solved only by comparing the course of evolution of each property, as laid down in the fossil record. It is obvious that the lack of substantial fossil record, and thus the inability to use that record to draw conclusions on genomic and phenotypic properties, have prevented the inductive derivation of genealogical lines. Furthermore, morphological complexity and comparative anatomy, extremely useful properties of eukaryotes for determining homologies, are absent in the morphologically and developmentally simple prokaryotes. As a consequence, a phylogenetic classification system only became available after the theoretical and methodological basis had been laid about 30 years ago. One of the main intellectual breakthroughs that helped microbiologists solve the problems of phylogeny. Scientists also commented that in the case of microorganisms, early evolutionary events can be documented only at the primary structural level of homologous and phylogenetically informative molecules. When comparing organisms, the number and composition of sequence differences between corresponding proteins and genes coding for rRNA reflect phylogenies and consequently allow the recognition of pairs of groups of organisms that originated from a common ancestor. Determining relatedness is based on sequence analysis of genes on their transcripts, also known as the semen tides. There are three categories: primary DNA, secondary RNA, and tertiary proteins, semen tides. Sequences of these molecules are molecular chronometers, records of evolution as they indirectly measure the time lapse since their origin, and the comparative analysis of primary structures is a powerful tool to measure evolutionary relationships. Episematic molecules to be used in comparative studies are synthesized under the control of tertiary semen tides, and above all is the chemical composition of cell constituents that have received considerable attention. Exogenous vitamins, phosphate ions, oxygen, viruses are not produced by the organisms themselves, and do not express any of the historic information that organisms contain. At any level of integration, the amount of history preserved will be the greater the complexity of the elements at that level, and the smaller the parts of elements that have to be affected to bring about significant change. Under favorable conditions of this kind, a recognition of many differences between two elements does not preclude the recognition of their similarity. The correctness of this hypothesis was demonstrated by the impressive phylogenetic trees of gene and protein sequences. Episematic molecules were not considered useful for deriving evolutionary conclusions because enzymes with different primary structures can lead to the synthesis of identical episematic or similar molecules in different organisms as long as the active enzymatic sites are similar. The two main tools, sequence analysis of the semen tides DNA, RNA, and proteins, and the hybridization of genomes for determining phylogenetic relationships in prokaryotes, were developed in the mid-1960s. Historically, the molecular approaches used were sequence analysis of proteins such as cytochrome C, fibrinopeptides and ferredoxin, as well as immunological approaches such as immunodiffusion and microcomplement fixation. However, the latter methods as well as protein sequencing became less significant with the availability of rapid sequencing techniques for DNA. The branching patterns based upon 16S rRNA and 16S rDNA sequences were surprising, mainly because these patterns showed that characters used traditionally to cluster organisms have in reality restricted phylogenetic meaning. Prominent examples of phenotypic characteristics shown not to circumscribe higher taxa in the past are now examples of characteristics whose evolutionary development has recently been or is soon to be unraveled. Today's sequencing of 16S rDNA in bacteriology is so widely accepted that it is considered a classical approach. The sequencing and analysis strategies and their bearing on evolution, phylogeny classification, and identification are now textbook knowledge. Good day, this is Professor Will, CBMD PhD on microbiology, bioinformatics, and computer science. Well, hope you enjoyed listening. Until next time, have a great day.